All right. Hello, everybody. Welcome to the Amass webinar today. Thank you for joining. With us here from the Amass side of things, we have, Jörn. You want to say a few words about yourself?
Yeah, sure. Yeah, my name is Jörn, and I'm working as a data and AI engineer at Amass. And I've been working at Novo Nordisk for the last 7 years prior to Amass, and before that at IBM Watson. So my entire professional career within life sciences and software. And yeah, I'm happy to present the new Amass course in this session.
Exactly. Thank you. Yeah, my name is Alex. I'm co-founder and CTO of Amass. Also worked in pharma at Novo Nordisk for some time, in health tech at Corti here locally in Copenhagen, but been really kind of building and doing research in the whole mining literature for science space for a long time. So excited to share with you today all the good things we've released over the last few weeks. It's really an accumulation of a lot of improvements that we've made to our platform, and specifically the data sources we have available. So really excited to share this with you all. And yeah, again, thanks for joining. We'll send out the recording afterwards as well. If you have any questions along the way, there's both a chat and a QA window. We'll try to keep an eye on it. Last time we actually ignored the QA section, so we'll do better this time around. Yes, so just to let you know what we have planned for today. So just a very, very quick intro why Amass is worth taking a look at when it comes to life science AI and what we've been building over the last 3 or so years. Then Jörn will take over, show you what the core architecture and our newest additions looks like that allow you actually to trace, starting from a gene in this one, in a few examples we have prepared, based on actually quite an exciting paper that came out recently as a case story around what you can do to go from a gene via then the mechanism of action to associated drugs, clinical trials, and regulatory documents, and what has been written in these regulatory documents. We'll show you along the way multiple things, how you can interact with these data sources, specifically the Amass MCP. We're going to demo this in Claude AI. It also works with ChatGPT and other platforms that support MCP connectors, together with an Amass skill that uses the Amass data sources, a quick Python script for the more computational amongst you, the more programming type, software types, and then also a very bespoke tool, but a tool that we essentially vibe coded, so very quickly made ourselves within 2-3 hours on top of the Amass API. That also brings the new data sources and the new cores to the front and makes them shine in a really interesting way. So yeah, just about, again, one slide on Amass as a whole before we jump into the story we have prepared for today. We've really, from the very beginning, and again, we've been building in this space for the last 10 or more years, more than 10 years, is that the number of papers, the number of trials, the number of regulatory approvals or documents, the number of drugs keeps on increasing, and it's just, it's only going one way, and that is upwards. And our mission for Amass in a lot of ways has been around making all this data more accessible, more findable for agents, AI models, chatbots to give you reliable answers. And today we're going to focus very much on the data layer, and also highlight why it makes sense to, or why that's kind of a prerequisite for anything in life science AI. In order to get reliable and useful answers, you need to have the data sources in place. So that's what we're going to show you here today. And then, yeah, I think I'll hand it over to you, Jörn.
Yeah.
And
let me share the stream. And can you see my slides?
Yeah, I'll just remove mine. Yes. Yep.
Perfect. Yeah. So yeah, welcome again. And I will kind of talk with Alex on the next 45 minutes about a paper that has been published this month in Nature, and kind of use it as a baseline to look some genes up that led to a drug, and also some that failed to become a drug, and kind of go via those examples to show you what you can do with Amass and our core architecture that I will also just explain in a second. And yeah, just show you what you can search for, how you can connect the data, or in this case, how AI is actually, for example, in the MCP layer, taking all this data together and extracting learnings very quickly. So I want to start with 4 humans, 4 people, and a gene called PRDM9. And this is kind of the textbook example of a gene that shows that if you knock this gene out, so for example, in a mice, you remove the gene, that it shows that those mice are going infertile. So they cannot get any newborns. And this has been, those 4 people in those study basically has shown something different, because this is a Nature publication about the Pakistan Genome Resource. And there are 4 humans that are actually having 2 to 7 children, each of them. So that kind of breaks the entire genetic dogma that has been in the textbooks for 14 years, especially on this gene. And that's why it's super interesting. So the Pakistan Genome Resource is a resource of 173,000 sequenced genomes from those individuals, 173,000. And this cohort is super interesting, because this cohort in Pakistan is quite common to have a marriage within cousins. And that's why it's way, way more likely that you get kind of mutations in the genome. I think it's about 1 million times more common that you get some kind of mutation in your genome compared to other cohorts. And this paper shows that there are a loss of function variants for about 6,500 genes. So this is almost 1/3 of all protein coding genes. So it's a really nice resource to get an idea of what a drug trial, or at least kind of experiments on mice or elsewhere, could have given you for data. So it's a super nice publication. And just to define a couple of terms also for maybe the non-biologists among us, more like the developer types, is that a loss of function variant is basically a spelling mistake that breaks a gene. So the gene no longer does its job. It cannot function anymore. And in humans, we have 2 copies of a gene. If you're healthy, born healthy, and have inherited those genes, of course, in a healthy way. And a human knockout is basically if both of those copies are missing, they're broken. So basically, the gene is effectively switched off for your entire life. And that is something that we want to do frequently within the lab, but also within in silico. So on the computer, kind of with data, trying to predict what would happen if we, for example, knock out a certain gene and what kind of effect it would have. And usually in mice and also in the in silico, you would kind of get a bit of a crash test data for this, because you give some kind of indication what would work or not. It's not one-to-one translatable. But this is super interesting in this cohort, that those individuals often are on average like 50 years of lifetime that they have had with those broken genes. So you can learn a lot about what kind of health benefits, for example, potentially does it have to knock out a certain gene, or what kind of consequences had, or also non-events, what is neutral if you knock out a gene. So I would rather trust this than any kind of in silico or mice analysis, and in terms of when it comes to knockout. So the old way, kind of usually when you read such paper in the afternoon, you're reading through the paper and find some interesting genes that you may want to follow up while reading it, because it may fit your project or your interest or your niche. Usually the workflow is then going to either Google or whatever and trying to, or more specified specific life sciences resources, just as you bring the gene and go to UniProt or to Ensemble, or you are then at some point maybe finding some crumbs, or that you could look up a drug on ChEMBL. Or the even more tricky part, you're trying to find some kind of clinical trials that are related to that. That is often tricky because of abbreviations or other kind of terms, and usually people are really good in also hiding those trials. Maybe you take it even further and look up what the regulator said, EMA, FDA, and the verdict, and then you're looking at PubMed, and you're kind of quite far in and spend a lot of time, and then you may still miss the most important part, that this drug, for example, was refused in one of the jurisdictions. So now the modern way with Amass, and also using the new technologies of AI and LLMs, we have kind of those 5 cores, public 5 cores that I will just talk about in a minute, that make this way more smooth. So for example, you can use our data and our retrieval stack that are optimized to finding this information in either our app or in your favorite AI assistant, for example, Claude or OpenAI, or directly in your code, either as a bioinformatician or as a developer building applications with your favorite type of language. So in the last webinar, Alex and I talked about 2 of our 5 cores, public cores that we have at the moment, and that was Biomed Core and TrialCore, which was mostly about scientific literature and clinical trials. And today the webinar was in the title about DrugCore and RegulatoryCore. I think it's self-explanatory, the names, but we also added recently just the GeneCore and released it publicly. So we included this also in this webinar, just to get kind of, don't hold back, and see also how you can connect all the 3 or even 5 to learn some interesting parts, look up information in a way that you couldn't before with also those cores together. And then let's go back to the study. So what the study shows on a very high level is that good drug targets are genes that you cannot break. So they kind of looked at all those genomes and looked at those knockouts, and they found that on average, the approved drug targets, the approved drug targets are 1.6 times more rarely that you will lose them if in this Pakistan population compared to all genes. So let's go through that. So each dot in this chart is kind of showing how often a kind of gene is found completely switched off in a living person in this specific cohort compared to all other genes. And they have kind of this bucket of, for example, the in vitro essential genes, and they are 25 times more rare to lose in the cohort. So it shows kind of that nature wants to, whatever we labeled in vitro essential genes, also in humans in this Pakistan cohort, you can see that they are the genes that are sticking around. Nature wants to persist them, keep them around. And the same with the mouse lethal genes category. It's 7 times more rarely that Pakistanis will kind of lose them over the when they get born in the specific cohort. And then again, for the approved drug targets, they're also rarer that they are losing it. So that means kind of on average, at least, that good drug targets are targets that of genes that are kind of essential to the human. But that isn't kind of good or bad by default. So the important part is, I think, in the study, or why it's so interesting, is that there is a human knockout in almost 6,500 genes. So it's something that we can look at those genes that has been knocked out in those persons and see kind of what the effect are. Are there kind of any benefits or disadvantages for those humans during their lifetime? And just before we kind of go and trace a couple of those genes, I just wanted to highlight an overall picture what we kind of can expect to find in those different stacks that we're having. So for example, if you look up something purely biologically, so for example, the target, information around the target PRD M9, you will only find something lighting up in kind of the GeneCore or PubMed core, because no one has tried to make a drug against this target. So you will not find something in DrugCore, RegulatoryCore, and probably also not TrialCore. Versus, for example, when someone has tried to develop a drug against it and maybe even went to the regulators about it, you will find information in all those cores. And we will explore that a bit more. But now back to Alex, and he will kind of walk us through how you can use our information stacks services with asking just plain English by using Claude.
Yes, awesome. Thank you for the layup and for the intro. Now we're going into one specific example. I'm just going to remove your screen from the stream right now. But you know, this is just one example of how the Amass MCP Model Context Protocol connector works in Claude AI. Again, similar things work in ChatGPT, where it's called an app, but also any other AI agent, actually. So now we have a question that is simply, I'm just going to copy it over first, go to a Claude session and just start it in here. We have an Amass skill that you can find on our website as well. That is called Amass API, which I'm not seeing here right now, which is a bit interesting. Let me maybe just refresh the window and start it here and see. Okay. A bit confused.
Can you look in your connectors?
Yeah, it should be a skill here. It's not.
Right.
Deactivated. Yeah. Okay. So it's the Amass API skill. Now I can also just show you, but you can also find this on GitHub or also on our website. We have different kind of skills, but this is a very general one, basically just explaining a language model, how the Amass API and the Amass data sources work. So if I'm gonna go in here now and yes, use it. So this using the slash commands, and then I'm just copying in the question. I'm gonna read it out afterwards. So it's, is the relaxin receptor drug seralexin approved? And what phase 3 trial backs it? And is its target RxFP1, loss of function tolerant in humans? So you can see now here that while I read it out loud, Claude was actually starting to do some work. You can see some of the reasoning here that is happening, but now it's actually starting to work with Amass. So you can see the Amass logo, and specifically Claude searched in the DrugCore, so to get specific information about approved drugs. So in this case, the query or the search was about seralexin, and then this is the result that came back from the Amass API, basically structured information about this drug, what it's doing, some alternative names, so some synonyms for this, trade names, and also maximum clinical stage that has been approved, that it has. You can see now that it's going on and searching for other DrugCore records while I was talking, for RegulatoryCore records or searching in regulatory documents, clinical trials. It is sometimes doing something that is called tool search. So it's looking for additional tools to use. And you can see here now that it's searching against trials, so pulling in more trials based on the NCT number and doing a lot of stuff to get a more fuller picture. So you can see here now it's still working and it's still looking across all the different cores that we just explained. I think the key thing here is that they're all connected and they're also all explained in a way to the language model via the MCP that allows an easy navigation across the different cores to come up with a concise answer to the question that I put in here. So I think this might take another minute or so. So I'll just go to a canned response to the same question that I ran yesterday to save the time here. But this is then the final answer. So the question was kind of, the prompt was 3 questions in one. Is it approved? And here says no, although the maximum clinical stage in the Amass DrugCore is approval, but it was also refused in this specific document here from the EMA. Jörn will come back to that in a minute and explain a little bit of what's going on there. That there's been a little bit of a discrepancy between approval in certain regions, but also refusal in, for example, by the EMA. In addition, you find a reference to the NCT number here, which is the key trial that was done. No, sorry, I think it was actually this one here, the RELAX-A HF2 trial, again with NCT number and a mass identifier. So this information comes from the TrialCore, and then some information about RXFP1 as the target gene here. We also provide additional information around that it's actually loss of function. Is the target RXFP1 loss of function tolerant in humans? Yes, it is. And then you get some additional evidence and some data behind that, kind of nicely summarized. So you can see here, just for this one example, we looked at DrugCore, we looked at RegulatoryCore, we looked at TrialCore, and it all was kind of part of the answer doing repetitive searches. And I think here I'll hand it back to you, Jörn, for now, and you can explain a little bit of what's going on behind the scenes and what these results mean.
Yep, absolutely. Let me get back to the presentation. You can see it, right? Just to make sure.
Oh, good.
Okay, good. Yeah. So yeah, thank you for the demo. So you could kind of see that how the MCP used the different cores that we're having and kind of stitching fetching the right information and kind of making a concise answer to the question. Then the way we designed our cores is that we kind of connect them together. So, and I just want to show that a bit more, because DrugCore, for example, is kind of a hub that connects a lot of those cores together. And you could actually have learned that by using also our API, you had all kind of those breadcrumbs already, the right evidence in one place to stitch this answer together. I will just show that in our platform documentation here, that you can see like how things are right now connected together. So for example, you can see that the DrugCore is connected to all the other cores. And that's what I meant also by being a hub. And then also how the others are linked together. If you're a developer, you can also click a bit here and then kind of get the different way how you can use the core architecture with kind of what kind of query params you could put in, the query, the search term in this case, and then other kind of query params that you can add to the API and get also an example of a response here. I wanted to show you a bit more the DrugCore. So kind of our cores are following all the same structure. So one endpoint for each of the cores is a search query that you can perform. One is a get record, which is if you have an Amass record, you can fetch the record for this Amass record. And one is always a lookup. And this lookup allows you to bring public data source identifiers and look them up in our system and resolve them to Amass IDs so that you can fetch uniquely without discrepancies the correct Amass record for this public identifier. And that way you can also integrate with your data that you're having already if you are having public identifiers with Amass. I wanted to go to the DrugCore search. And one thing that we are allowing here in the search, you can see it already. I just wanted to say, like, you can either look for keywords, for example, a drug name, but you could also look, for example, specific mechanisms of actions that are searchable. So you don't have to know already in the beforehand what you're looking for, but you could also look more free text as a query and find connected information. So for example, seralexin, that was, if I, for example, let's take the gene. We kind of come from the paper, we have the gene, RXFP1, and search with that, then we get this record to seralexin. And that was also what the MCP has found. And it says, max clinical stage is approved. I'll get back to that. And then the mechanism of action, it is an activator for two receptors, the seralexin receptor 1 and 2. And you can, in addition, now let's say you're searching for something else, you can also kind of filter by the type of drug. For example, if you're interested in small molecule or whatever you can see on the list right now. And the same for kind of the clinical stage, what stage you're interested in. And then the last one, that is what I wanted to show you with the hub, that is, for example, where you can include those references to the other cores. So for example, I could check off, just give me all of them and give me the connections to the other cores. And you can see that there, for example, those 16 trials, one publication related to this, then a RegulatoryCore document, and those are the two gene cores, the receptor 1 and 2. And the same I would like to show you for the GeneCore. So if we're searching here for the gene again, we can find it here with its symbol, its name, the synonyms, which we're also enabling you to search in easily, the summary. And then we have all kind of public identifiers that you could either kind of, as I said, come with and resolve to ours, but you can also search it that way and find it on our API. And then also if you're using that in, for example, building an app, you can kind of link out or fetch additional information from those other data sources if you would like to. Again, we can set certain filters. The gene types are supported here. If it's druggable or not. So we have a lot of target intelligence that you can search by if you don't come, for example, directly by the gene or the symbol or whatsoever. But if you're more like exploratory, then you can add easily those filters to find target intelligence and filter, for example, your targets down or find potential targets. So for example, we have the tractability here of the small molecule antibody and other clinical and project. And we have also the genome AD constraint. And this one, for example, is interesting. Here's the loss of function again. And those are the kind of key values to look at. For example, this also shows that this is, if this is below 0.6 in this version of genome AD, then it's likely to cause a disease. You can see it's not, and this also goes back to the story. You find a filter about if it's essential or not. Again, you can include other references. Now we have the reference back here to the DrugCore. We have also more addition on the protein if you're interested in this. If you're, for example, a chemist and would like to learn more about the structure and its chemical properties, we kind of also support that in the API easily. Let me go back to the presentation. Yeah, let's do the presentation first. So yeah, this is basically the DrugCore API that I just showed. So it just resolved with the other cores to show you this information here, the gene targets, the trials. That was also the one that in the MCP was highlighted. And I will get back to that, why this is an interesting study. And then kind of the regulatory, what the EMA said. Maybe I can, let me actually go back and show that to you. So if we look in the RegulatoryCore and look here, for example, seralexin, how do I spell it? I should find it in here. So this is, for example, from the EMA, and it showed that the authorization status is refused. And in addition to that, you can see kind of the therapeutic indication that was for treatment of acute heart failure by Novartis. And in addition to that, you can also see all the document sections. So the way those cores are working is that we have records for the agency of FDA and EMA, And then we have the current status of what the status of this drug application is at this very moment in time. Plus, we have the documents of the, for example, for EMA, you can see the EPAR documents, or for FDA, the reviews and also the label information. So you can see then those document sections, and if you, we're supporting searching in all of these at once, so you don't need to come by, for example, the active substance or the name. You can really search free text across document sections, the raw PDF basically from the EPA. One last thing, and maybe also just to show you the filters here. So you can filter down by molecule type, by authorization status, and I will show that a bit later, how to use that a bit more explorative. If it has an orphan status, if it has a designations. And then you can also specifically, for example, in a certain Amass ID, so let's say you have found the record that you're interested in, you kind of can lock it down to the specific record and then search just specifically with this query now within this record. And again, like we have to include other kind of references and details that I will not show at the moment. I also want to show you that you can look up specific documents. I actually do that later. Now I'm getting too far ahead of myself. Let's go back here and talk about what you've seen, what the MCP showed, but what actually also the API just showed. So there was a bit of a discrepancy. Initially, it looks like at least the discrepancies, the kind of two answers. There was like, was this drug approved? And seralexin is approved as a drug, but it's in a minor jurisdiction, and it's actually in Russia, versus the EMA have judged that in 2014 to refuse it. And we can actually also see in our API, and also the MCP can kind of, if you drill down and want to figure out why it has been rejected, we can look that up in the API and search for it. So one way, now I'm going back to the API reference. So one way is to search in those sections, and then you can extract for each of the sections that you're, for example, interested in. Let's take it as example, this specific section about the discussion on clinical safety, which is in the EMA-EPA document. You can get the document section. That's another endpoint. And you can, for example, put the Amass ID for this drug application up here, and then the document section ID that was in the other API call up here. And now you can get the content for this specific section. Again, this is the discussion on clinical safety, and then you get kind of the full content of what has been talked about or what has been written in this section. And it's not really nice to read it in here in this API reference. So that's why I built for our exploration of this drill down here a little Python script. I will not go too much into the Python script. I will just explain it while running it, what is happening. So if we zoom a bit up, you can see that we are for the RegulatoryCore. Let me zoom in a bit so maybe it's easier to see. For the RegulatoryCore API, we're looking up the record for seralexin, the active substance, and then the name for the drug, olesarzan. And then again, this is just what we looked at in the API reference in the UI from Novartis and the document sections here. And now specifically, I'm pulling out two document sections. So the first one is the benefit risk balance, and the third section. And there kind of, it has 26,000 characters. So that's why I didn't want to read it in the UI. It's a bit awkward. And also here, I just pulled out the most important part. And it says, in this dossier, these conditions are not met and confirmatory data are needed to support evidence of clinical benefit. That was kind of the first assessment. And then also in the other section, in the divergent position, you can read that we believe that the applicant has addressed the ground for refusal, blah, 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 sufficiently. while allowing a recommendation for conditional approval. So the EMA has approved seralexin renaissance conditionally, and they wanted to see more confirmatory data for the effects of the drug working on heart failure. And basically, Novartis ran it, the confirmatory trial, on 6,600 patients. And that was also what Alex showed in the MCP. I can't recall the name of the study, but basically it missed to show the primary outcome. So it missed that, and that's why it got refused by EMA. And the interesting thing is now that you could have seen that, or you can see that in this Pakistan cohort. There are actually 16 people between 30 and 73 who are having this gene knocked out, and they don't have any consistent deficiencies. So it's very likely that the target, with this knowledge, that the target couldn't have delivered. And you could also have seen that in the genome AD constraint that I showed in the API for the target, that it was basically such a high value that it was not likely causing disease, and it was also not essential. But again, to be fair, you couldn't have known that in 2014 because the cohort, the Pakistan cohort, didn't exist at that point in time. So there was no way to kind of see this data here. And also the genome AD constraint was also not published. This method was not available back in time. So you couldn't have killed it now, but back in time, but now you could have had if you would have looked up those data sources, which I think are interested. So just to sum that up, what went wrong here? So Novartis tried to kind of crank up a receptor that actually nature showed that you could live without with. So it was really now in retrospective, at least, unlikely that this could have a benefit to the human condition. And we know that now. But that doesn't mean that now every kind of knockout that you see in this Pakistan cohort, that this is a bad target. It could be also the other way around. So there are definitely also targets that show benefits on human disease or human condition. And Alex will show you the next one, which is APOC3.
Yes, thank you. I'll go over here. And so we have one more example here to show when using the MCP Live. Again, using APOC3 antisense drug olesarzan. Is it approved? And what does it do to the gene? And is loss of APOC3 tolerated or even beneficial in humans? So I'm just going to copy and paste this to a new Claude session here. This time I can use the skill right away. I'm going to copy in the question. Again, the same thing is going to happen like what we just looked at. So looking at the different cores. So I'll just go over to what the answer looks like when I ran this question earlier. And you can see here that, yes, it is approved. Here it's marketed as Tringolza, and then approved by the FDA in 24, for example. What does it do to the gene? There's different explanations here. Information that is actually taken from the FDA label. I'll come back to that. And also we actually, in this case, we also link it to literature. So you can see here that we also have specific PubMed links, for example, that explain more about why this is a beneficial loss of function mutation actually in humans, along with kind of three key papers for it. I also then asked a follow-up question. So what I did, just to make sure that also to show how you can go deeper in this. So what I did is kind of ask for click the reply button here and then ask, okay, what does it say about the mechanism of action? And again, I'm referencing the FDA label here. So now Claude went into Amass, looked up the corresponding label information, then showed me directly what is actually shown on the FDA label. And then in the specific sections here, so in section 12.1, it talks about this, and in 12.2, it talks about this. Just to make sure, again, also you get a link here. So if I'm looking at this document, it's actually inside the PDF somewhere. So this is the label from the FDA for this drug, and then under section 12.1 and 12.2, you basically get the information that you can, you could have found it yourself. So it probably takes you at least half an hour if you know what you're looking for. If you don't know what you're looking for, much longer, to just like find this very specific piece of information. And then, it's really nice that this is also just available with a prompt away or a question away here. And of course, behind the API as well. So the last thing that I want to show here in terms of demo is additional applications on top that you can build on top of the API. So this is more if you're building tools for yourself or maybe for your team. What can you get out of the data directly as well? Jan already showed you what the data looks like, how the data is structured, but having that structuredness around the data actually means that you can build very nice dashboards and applications on top of this. So we have now I'm under platform.amass.tech, which is our developer platform. We showed this last time as well. You can get an API key, you can get credits, and most importantly, we added an app gallery, which we haven't here shown on the stream or in the webinar yet. So feel free to take a look at it. We're now gonna just look at the first example application here that we have built using the Amass API. Took us maybe 2 hours to get to this, so you can actually build these very, very quickly and iterate to build your very bespoke software to solve exactly the problem that you're having here. So what this is doing, it's called From Gene to Approval. I can just open it here. So you have a little bit of an explanation. What is this doing? You have an explanation of how we build it with a very specific prompt that we used here in a software called Lovable, which is basically a platform that allows you to build bespoke software solutions yourself. And this is what the result looks like. So it is basically allows you to link certain genes to drugs. So I can now here come in, I search for HBB. I'll come back to that in a minute, why that's relevant. And you can see here for the HBB gene, we have actually approved drugs here. I can come and look at PCSK9, of course, super relevant target these days, or for quite a while now, where I can see that, okay, it's actually very popular, being studied both in preclinical, phase 1, phase 2, phase 3, and targeted by these antibodies, for example. And then get a whole lot more information around what are these drugs, what is the modality, what are the trials here that can kind of very quickly go to say, okay, what is the NCT number? And I guess the point here also, right, if I wanted to have specific other information that is shown here, wanted to massage the data in different ways, visualize it, it can do it very quickly. The key is the data, and the key here comes from the Amass API, where we have the different cores that we can connect in very interesting ways to enable these kinds of applications. Also, of course, reach out if you want to know more about this. We'd also be happy to discuss this offline and how that works. And then so just to come back to the presentation here, again, based on the Claude response, we saw that it is actually in APOC3, there's approved drugs from both the FDA and the EMA, and the inhibition is actually a beneficial inhibition that can protect you in different ways. So this is again a paper from the Pakistan cohort study that we used as an example here. What you can see here is that for APOC3, you actually see a lowering of triglycerides as a blood biomarker in the population they looked at. So it's also very pronounced, 0.9 standard deviation lower with people that have lost, I think, both copies of APOC3. In this case, same for PCSK9, looking at LDL cholesterol, and HBB is also shown. That was the other gene I just showed in the trial in the application here, where the LDL cholesterol effect isn't as pronounced as for PCSK9, but still a modest drop. So you can see here it all ties nicely together, both in terms of what is shown in the study, but also, of course, in terms of the information that you can get from the different cores in Amass.
Yep, thank you for that. I'll take over the stream again. All right. So I would like to show you also another way how to use the core. So you don't need to know already the gene beforehand or the active substance or any kind of drug name, because in our course, we are searching in many fields, so which are not only synonyms or names, but also kind of summaries and for regulatory and the entire kind of PDFs. So what you can do is, for example, being very explorative and using the query params, or we can call it filters, in your calls to the API, and also the MCP can do that. So basically, if we want to see which drugs have been refused or withdrawn for heart failure, we can use the API again. In this case, search, put in heart failure, and then go to the authorization status and say anything refused, or let's say withdrawn voluntarily or withdrawn forced, and see what comes back. So for example, then top hit again is seralexin because of the acute heart failure. That's also why I use it as an example. But you can also see kind of others here, for example, from Amgen, Colonor, and the other one, I think there's one tablet at least. There you can see kind of in the designations, there was a priority review from the FDA for this one. And you can also see that their authorization by agency, there's another one. And this is the identifier for the first one here. And I haven't read too much what the differences are, but I guess maybe one is tablet, maybe in another form, at least 2 applications of these. And yeah, you can find a couple of other ones, Kinato, in here. So you can use those APIs to be very generic, to search them, and that's also basically what the MCP does. And it gives you more control if you kind of know what you're interested in. Maybe you're interested in a certain authorization date range, or you're interested in a specific molecule type. So that kind of helps. And I kind of created with this like the bit of a quick overview of a heart failure graveyard over drugs that were rejected, just to show you another way to use it. And then also the other way around, that was also what Alex showed. If you're looking for APOC3, you could get to this view by combining DrugCore and RegulatoryCore, because you can search for the mechanism of action with APOC3, and then you can find those drugs and look up in regulatory if they got approved or not. The same with the app in Lovable, you can see this. So, and then to lift it up, make it more generic, there are different kind of questions and how you combine those cores, and you can combine them all together. But basically, what has been tried on my gene? If you have a list of genes, or I want to prioritize my genes by using target intelligence, you can kind of use target core and then GeneCore, and combine it with DrugCore, or directly search in DrugCore if you know already the gene name here. Or you can go who approved it and where, looking at the regulatory evidence. And then you can also, for example, make comparisons. So where do FDA and EMA disagree? And we also actually have a little app for that in the app gallery, which is called the Transatlantic Divergence Stack. So that is kind of using exactly that. If you search for a certain molecule or drug, or again, any kind of free text search, you can quickly filter by the agency, and you can simply compare that this, I think, molecule, for example, has been approved by the FDA but refused by the EMA. One final gene in this Pakistan cohort is LRRK2, which is super interesting. It is kind of a hot target right now in Pakistan. So this cohort, the Pakistan cohort, shows that there are actually 2 knockouts, and both of those knockouts, they show that they have some kind of early kidney disease. So there may be something to pursue, but you may want to be careful with kidneys. So if you look up RegulatoryCore, there's nothing yet. But there's still something, I tried to search a bit, and there's still something in flight that you can find if we are looking for, let's look at the DrugCore. And I couldn't find the mechanism of action with this, but what I could find, and I thought it was interesting, or that I could find a Merck inhibitor. So I just copy the name and the synonym, and then let's try in TrialCore to search for the name and synonyms. That would, oh no, let's do it again. Apologies. Okay. So let's see what comes up. There was something that I've seen here about, Parkinson disease. Beside Parkinson disease, there was observational condition, Parkinson disease, and then also the keyword that the gene is mentioned. And I also filtered a bit on phase, so actually tried to phase 2. I think I even got some kind of findings that was Biogen and Denali, Parkinson disease. Yeah.
Just a quick note. We have a question also from the audience. Maybe we should kind of wrap up the presentation just to make sure that we have a little bit of time for that.
Absolutely. Yeah. Awesome. All right. Time is a lot. Let me go back to just the overview. So basically what we discussed over the last 40 minutes were like 5 different targets, 5 different verdicts. I will not recap it again, but some of them kind of were positive signals, some of them negative signals from the trial, from the paper, which were interesting to follow. And that was also the final one. You can use us, as we also explained before, either via our app as a non-technical users, where you can use the cores and add your own information and search and run workflows, which we call templates, kind of repetitive, and you can run different targets through. You can use us via the MCP. We just showed that. Or if you're a developer or want to develop something internally, use the API. Yes. And with that,
awesome.
Yeah, let's go to questions.
Cool. We leave this on and we go to this view. I think that makes most. Awesome. Thank you very much also for everyone for sticking around. There was a question here. I'll just add this to the stream. Let's see if that works. Is the data being returned from a static database or being accessed from the source URLs live on request? If the latter, are there occasions the values are not correct? This comes from Joe. So thank you, Joe, for the question. Do you want to take that?
Yeah, absolutely. So each of our cores is kind of composed of different data sources, and we are kind of normalizing them and kind of curating them nicely up. And all of those, or most of the cores, are daily ingested, those data sets, and kind of keeping up to date. And we're not using the live public available data, like we don't have hardware it directly, but we have our own data repository, and that allows us to optimize how you and the LLMs, LAI, basically can search in those stacks. So we have kind of evaluations and optimized that you can naturally, with natural language, search against the data. So we have our own search index, basically, and curated the search index to make this super efficient.
Yes. Maybe also just a note, I think by now we have probably more than 50 million different documents, pieces of information in the database. It's actually quite a large amount of data, and each of the pipelines that you mentioned that is running, most of them daily, is actually also pulling information from 5 to 6 different sources. So it's quite a lot of different orchestration, handling, merging, harmonizing things we need to be doing in the background. But of course, it is a service that we're providing here with the API, and then kind of the main idea is really to save the data scientists, data engineers out there from doing that yourselves and doing all that work, but making it available such that you can actually, focus on the new exciting biology that maybe your biotech company is working on, and kind of get that, get in front of your team quicker. There was another question that just came in. What are additional data sources cores you're planning to add in the future that could be relevant to extend to the use cases you presented? It's a very good question. I'll just add it to the stream as well. Do you know that by heart, Jörn, or should we pull up the roadmap, or do you want to answer that?
We could pull up the roadmap. Do you have it? Or I can find it.
Yeah, it's platform and then and the documentation.
Yes.
Yes.
Yeah. So one of the things that we're working on is enriching the clinical trials to have international clinical trials. We are also working on for the trials to have structured readouts, biomarkers, and so forth. And we're planning to add soon patents, probably in beta, right? Do you want to say a couple of things to that?
Yeah, they're very close to releasing the first batch of patents. Also linking that to the other cores, so they will be available soon. Probably in a preview mode in the beginning, but yeah, reach out if you want to know more. Also in general to the roadmap as a whole, of course. Very open to have feedback from you, all of you that are using it. Very happy to also reprioritize everything we're showing here according to your needs. Yeah,
yep,
so that's the picture.
Absolutely. And then we also want to, of course, not only expand on the links that I showed originally between the cores, but also enrich kind of the depth of it, making it even better to pick up the relationships even better. So that is something ongoing that we do all the time.
Yes. And of course, optimizing for quality, performance, and cost. Yeah. It's always on a roadmap. We're also publishing some of those and some of the findings we have and benchmarks on our resources to share that with the world.
Yeah.
Yeah. I think that is it. We're right on time. Thank you, everyone, for joining. I think this has been a very nice session. Any last words?
No, thank you so much for your attention and joining us and being curious.
Let's remove it from the stream. Okay, cool. Thank you all.
Good.
See you next time.
Thank you.
And have a good summer.
Bye-bye.
Bye.